So we've kind of coined the term seamless design within the FDA to refer to this. So historically, the phases of drug development were very discrete, and they had very particular goals and kind of characteristics of the nature of the trials that were done. So phase 1 trials, or first-in-human trials, were trials that were done in small numbers of patients, generally not selected by particular tumor type. They were often patients with advanced solid tumors, for example, whose tumors were refractory to all available therapy. And the goal of them was to start at a low dose that we thought would be safe and escalate up the dose until we got to the point that we thought that the toxicity precluded going any higher. And so the goal of those studies, which were small, they often enrolled maybe 30 patients, was to select the right dose for further study and to identify the safety issues with the drug, building on the animal studies that had previously been done. And then the drug would go into phase 2, and phase 2 would sort of be an early look at efficacy. So it would be saying, okay, in phase 1, maybe we had 30 patients and maybe there were only 2 responses, but both the responses were in patients with HER2-positive breast cancer. So in the phase 2, we're going to narrow our focus, we're going to limit our enrollment to patients with that disease, and we're going to try to get a sense of how much activity this drug has. And that was often assessed with response rate, sometimes in a randomized trial, but usually in a single-arm trial. And again, pretty small, maybe 50 patients, maybe 100. And then if there seemed to be sufficient activity in terms of response rate, then that's when the question would be asked of whether the drug would move on to phase 3, which would be a randomized comparison against the best available therapy, either sometimes as a separate and new option, or sometimes as an add-on design where one arm got the best available therapy and the other arm got that same therapy to which the new drug was added. And the goal there would be to say, does this represent an advance over the way we're currently treating this disease? And that phase 3 trial would serve as the basis for FDA approval. Part of the reason that the drug development system was set up that way was that most of the drugs were destined to fail, because we were starting in phase 1 with unselected patient populations, and the likelihood that we would see a response going in these patient populations with absolutely no selection for the types of tumors that might respond, no biomarker selection, and treating patients whose tumors were already very refractory, so probably have a lot of mutations and other changes that may result in resistance to treatment broadly, sort of set those drugs up to fail. As we've gotten better at developing drugs that are targeted to specific abnormalities in the tumor, we've been able to take those first-in-human trials and do them in patients whose tumors are less refractory, patients who may have had even just one line of therapy, for example, but who are biomarker selected. So we've enriched those very early studies for the sorts of patients that we hypothesize are going to be the ones most likely to benefit. And when we do that, we still are able to get the same information we've always tried to get about dose and about what side effects are going to be an issue with the drug, but we also get a sense of how much efficacy this drug is going to have. And in some cases, the amount of efficacy that we observe has been pretty astonishing, and it has made us not want to do this traditional phased approach, which introduces a lot of delay. When you complete a phase 1 study and analyze it, and then now you're going to design the phase 2, and you're going to negotiate contracts to open it, and then you're going to analyze that, and you're going to design the phase 3 and choose all your investigators. There are a lot of inherent delays in that process, so that process would routinely take a decade or more from first dosing to getting the drug approved. So what we have seen happening, and this started a few years ago with pembrolizumab, the first immune checkpoint inhibitor, to use this kind of approach, there was a lot of efficacy observed in the first-in-human study in melanoma and non-small cell lung cancer, and therefore, rather than doing this traditional discrete phase development, they simply decided to modify the protocol, amend the protocol, and add additional cohorts to look at efficacy, to refine the dose, to get greater numbers of patients to assess the safety. And in effect, they did really a whole drug development program within the context of their first-in-human trial that ultimately enrolled well over 1,000 patients. So there's been a move by pharmaceutical companies to try to replicate that model for obvious reasons. It's a huge time savings, and that means their drug is on the market sooner. We have been encouraging this for appropriately selected drugs. So Dr. Pazder and I wrote a perspective article in the New England Journal about this, and we have subsequently worked on a guidance that will be appearing from FDA at some point in 2018 to basically help companies identify the sorts of drugs that are appropriate to do a seamless design for. I think at this point the soundbite message would be that drugs that are capable of qualifying for a breakthrough therapy designation are the kinds of drugs we would like to see targeted to these programs. The efficiencies there are best utilized with the drugs that have the most promise, and I think that the concerns that we have potentially with these models are mitigated when we have a drug that we think is going to be really effective. One of the problems with doing an entire drug development program in the context of a single first-in-human study is that we don't have those scheduled pauses. We don't have those periods in between each trial for everyone to sort of get together, look at the data, think about it, say, what does this mean? How does this impact the design of the next trial? These seamless designs move very fast. And the other thing with the seamless designs is that because they are expansions of first-in-human trials, sometimes you have very limited or maybe even no true control arm data. These are, in many cases, effectively a single-arm study in the sense that all the patients are getting the new therapy, and that makes safety very difficult to interpret in them. If you don't have a control arm to be able to say, oh, well, heart attacks occurred in patients who are getting the control therapy too. Maybe this is a consequence of their patient's disease. Maybe this is not a consequence of the therapy we're giving. If we don't have that control arm data, it becomes really tough to interpret safety signals we see when we receive those data on the back end as regulators to know, is this a real safety signal or is this just a part of the natural history of this disease? So the bottom line is that I think seamless designs are very efficient. I think that they represent the best kind of clinical trials for drugs that truly have unprecedented levels of efficacy. They increase access to study drugs for patients. They develop the drugs more quickly. They minimize randomization of patients to control arms that are probably going to be inferior, but it has to be appropriately selected drugs for it to make sense.